Introduce tonight's speaker. Dr. Ana Aguilera is an associate assistant professor at Simmons College here, or not here, but in Boston. She is a UMass graduate. She got her master's from Amherst and got her doctorate in Boston. She's also on the board of directors for Massachusetts Rivers Alliance. She's been in academia for quite some time, and all in biology and ecology associated with invasives primarily. The title of her talk this evening is Differential Decomposition of Acer platinoides in Acer saccharum, Effects on Decomposition Rate in Macroinvertebrate Community Composition. Please welcome Dr. Ana Aguilera.
Thank you. I'm really pleased to be here. You guys are such a fun group. I have, as Brett said, I'm talking about Acer platinoides and Acer saccharum, so Norway maple and sugar maple. And this work, I'll go through. I have Cinzia's name. This is my student, Cinzia Valentine. She's a recent graduate of Simmons College. An excellent student, and most of this work is spearheaded by her under my guidance in my lab. So I can't speak highly enough about Cinzia, and I don't want to take any credit from her because she was really driven and really excellent on this project. Let's see if I can. Is it the red one?
No.
Oops, work it off. Okay.
Right and left.
Okay. Right and left. Okay, so this project, I wanted to introduce another plug, not just for Cinzia, but for ERIN, which is the Ecological Research Education Network. This is a group of people I work with. The mission is to create a model for collaborative ecological research that generates high-quality, publishable data involving undergraduate students and faculty at primarily undergraduate institutions. So Simmons College, we're a women's-centered college on the Fenway in Boston. Biology department has no graduate students, so all the work in my lab is done by undergraduates. And that can be really challenging, as a lot of you probably know, to get publishable data. So what ERIN does, you might have a brilliant idea, and I say, oh, I'm going to be the PI of this project. You write up the methods, and you submit it to the group, and people all across the country at similar-sized schools with undergraduates get involved. So we get these large data sets, and we can publish interesting papers that have broader conclusions, because it's not just my undergraduate study happening on the Muddy River in Boston. So this is a really, really, really great organization. I can't speak highly enough about this. The specific project that Cinzia and I were involved in is called DATIS, Decomposition in Aquatic and Terrestrial Invaded Systems. The PIs for this are Tracy Gartner, she's at Carthage College, and Carolyn Thomas, she's at Ferrum College. So we've jumped in on that with them, and they were interested in how decomposition of invasive species is different in aquatic and terrestrial systems, right? They, sort of the scientific background for this is the facts that woody invasive species influence community dynamics and ecosystem functions, right? In what ways? They can influence nitrogen concentrations in the soil, decay rates are often higher for invasive species. So if they're decaying faster, then nutrients are becoming available faster for local, native, and rare plants. And so those sorts of shifts in ecosystems can really facilitate invasion and harm native plants that have not evolved under those conditions, right? Oops, sorry. The impacts of invasive species, I don't know what I'm doing, I put it down. Impacts, I don't use one when I teach, so I get excited to have it, but I don't think I should use it. So impacts of invasive species vary with the invasive species, right? It's really hard to make broad statements. Invasive species do this. Of course, they don't all do the same thing because they are incredibly diverse among themselves. And their impacts can vary with their own relative abundance. So you might have a teeny patch of garlic mustard is going to have a much different impact from a large monoculture of that same plant. It also varies with the species interactions around it. So depending what system is being invaded, the neighboring species are going to alter decay rates or make them faster, right? So this is just a very complicated picture, right? You can't make a general statement. And then when you put that into a broader context of the different places, in this country and obviously worldwide where things are invasive, you know, weather conditions are different, and that's going to influence decomposition rates. This shows U.S. soil nitrogen, with the darker areas having higher concentrations of nitrogen, and the nitrogen content of the soil is going to alter decomposition rates, right? So there's, you know, the bedrock is going to alter these things, right? So it's very complicated, which is sort of the beauty of doing this with Aaron, is that we can do the similar study, the same study in a lot of different places, and try and see if there's any broad patterns. This shows the locations that have participated in this project, with the little teardrop missing for me, which is sad. I didn't put my own teardrop in. So we've also are doing this in Boston. But so this decomposition study is taking place all over a lot of different schools across the country. So it's really fun. We're getting a ton of data. So, oops, this was supposed to be animated, but the red line's going through there. So the questions, well, the crossed out questions are the questions that the DATIS project is examining, right? My student, Chintzia, and I contributed data to this, but she also asked additional questions. And these are the results I'm going to go through with you guys today, which is why I crossed off the top two. But I wanted you to know the specific questions of the main project as well. So Tracy and Carolyn are asking, are decomposition rates of invasive species faster than native species across a range of environments? That's been shown a lot of times individually in different environments. We did this large study to see if we can see a broad pattern of that, if that holds true when you do it in a range of environments. Does increasing relative abundance of the invasive have a linear impact on decay rate? So looking at, you know, if there's concentration of litter in the environment, does that have a linear impact as you increase in concentration of the non-native species? So if you're curious about this, you'll have to stay tuned. We are working on analyzing all that data, and that'll be published hopefully shortly. But what my student Chintzia, she was just did this in Boston in the Arnold Arboretum, right? So her specific questions for her smaller subset of this is, does replacement of Acer saccharum by Acer platanoides alter decomposition rates? And does replacement of Acer saccharum by Acer platanoides alter diversity and abundance of macroinvertebrates? So I am going to do a lot of talking actually about invertebrates today, not just plants, because she found some cool stuff. Okay, so the design for the larger experiment, leaves were collected from locally relevant invasive and morphologically functionally similar native species. So depending on where you are in this larger DATIS project, which species might be appropriate for you. So the large project is comparing buckthorn and black cherry or gray dogwood, honeysuckle to spicebush, privet to Texas mountain laurel, tree of heaven to smooth sumac, and Norway maple, sugar maple, or red maple. So in the larger Aaron network, people are working on all these species, and Chintzia and I did Norway maple and sugar maple. So a little bit of tiny bit of background on these two species. Here's a picture of Norway maple and a picture of sugar maple. Acer platanoides, the Norway maple, as I'm sure most of you guys know, is a fast-growing, hardy, and shade-tolerant tree. And sugar maple is these things too, but it's been shown that Norway maple is even more shade-tolerant than, Norway maple is even more shade-tolerant than sugar maple. That's one of the advantages it has over sugar maple. It also has a later senescence, so it has a little bit of a longer growing season. It definitely senesces later in the season than Acer saccharum. And there's work that shows that soils that are invaded by Acer platanoides reduce the growth of Acer rubrum. So they have been shown to reduce red maple growth, just the soil. So if you grow red maple seedlings in soil that's been trained by Norway maple, those seedlings grow more poorly than if they were in their own soil. So there's a bunch of stuff happening here. So we did a litter bag study. So we constructed litter bags. Litter bags are mesh bags that you put the leaf litter into. We had 5 separate treatments, and they varied in their invasive abundance. So in one treatment, there was all native litter. So you collect litter in the fall, and I put 8 grams of all sugar maple into a litter bag, right? My other treatment is 25% invasive litter. So 3/4 of that bag is sugar maple, 1/4 is Norway maple, right? So then 50%, you get 50-50 both, and then you get 75% of your invasive species in a treatment, and then all invasive species. So if you can picture it, I've got these 5, I'll show you a picture. These are the litter bags, right, mesh on each side. So this stack would be all 5 of the same treatment, right? We replicated this 5 times in the field and at 2 harvest times. So to try and get an idea of decomposition rate. So you put out the litter bag, you have 5 separate sites, and you pull them out over time. You pull them out once and you weigh what's left. You pull them out a second time months later, weigh what's left, and the mass lost gives you decomposition rate. It's very low tech. It's a really great project for undergraduates because there's not a lot of technical stuff going on. So, and we did this in aquatic and in terrestrial sites. So the construction of the litter bag, they are 20 centimeters square. The bottom mesh is bigger than the top mesh. So this is important for Cincia's edition of looking at invertebrates, because you put these litter bags big mesh down, right? And the big mesh allows for invertebrates to crawl in. So they serve as tools to measure decomposition rate, but they're also conveniently macroinvertebrate traps. When you go to pull them up and get your litter and weigh your litter, you can also open them up in the lab, dump them out, and they're crawling with invertebrates. And so you can trap the invertebrates that are drawn to that litter. It's really, really fun, actually. We also made 15 travel bags because we drove these bags back and forth to the field. And the travel bag just accounts for all the dry litter that's crumbling and flying out and ending up in your car instead of ending up, in the stream or in the ground. I briefly described how we calculate decomposition rate just by looking at mass lost over time by pulling out sequential litter bags. What did we do with the macroinvertebrates? We took total abundance. Cincia calculated species richness, or she counted species richness. She calculated Shannon-Wiener diversity, and also looked at Jacquard's coefficient, Deckard's coefficient. And this is a picture that shows the types of stuff. Oh my gosh, sorry, I keep doing that. We found. There's midges, there's stoneflies, dragonfly larvae, mayflies, lots of nice, gross, creepy, crawly stuff that make lab work really fun, I think. So just to refresh everyone's memory on some of these indices, the Shannon-Wiener diversity index, so richness is the number of different species you have, right? And there's these measures of diversity that look at species richness, but also evenness, right? And that's an important concept for diversity. And so this here is the equation to calculate Shannon-Wiener diversity, where P is the proportion of individuals of one particular species found, little n, divided by the total number of individuals found, large N, right? And then you have S as the number of species. So you sum it from 1 to the total number of species with this proportion of, I found 2 stoneflies in my pool of 300 insects, right? So 2 would be the little n, 300 would be the large N. You're taking the natural log, and then summing that for each one, right? And if you have a high diversity, then you've got some evenness, you don't have 299 midges and 1 stonefly, right? That wouldn't, that 1 stonefly doesn't count for as much when you do these diversity indices. Jacquard's coefficient, which I was less familiar with, but Cincia was really excited to do, and it was very neat, looks at similarity. So she looked at Jacquard's coefficient between pairwise combinations of the same treatment. And how that works, it's always pairwise, you're always comparing 2 bags, comparing the bags, where A is the number of species common to both litter bags of a given treatment. B is the number of species unique to the first, and C is the number of species unique to the second. So by calculating this coefficient, the higher the number here, right? Imagine if they all are the same, you're going to get 3 over 3 plus 0 plus 0, right? A 1. So it's going to range between 0 and 1. And as you get a smaller fraction, you have the 2 bags to be less similar, right? So we have these 2 indices, how to look at macroinvertebrate diversity. We put these bags in the Arnold Arboretum, which I'm sure you guys are familiar with. This is the Bussy Brook. And for those of you that aren't super familiar with it. This is this side is uncurated, unmanaged land here. So it's pretty wild. I took Cinzia in there, and it was so funny because it was one of our first field sites, first field trips, and I managed to get myself, like my hair got wrapped in some thorns, and my foot I got completely tangled, and I hit the deck, and I had to ask her to come untangle me. So that's why I like to break the ice with students that might be nervous with me, and just like make a total mess of myself. So we've had a lot of fun running around in this unmanicured part of the Arboretum. She's a great person, and she did save me that day. And then we had 2 sites here in the mowed, manicured area of the Arboretum. And so this is what our terrestrial litterbags look like. They're right outside the stream. Again, so there's 5 treatments, 1, 2, 3, 4, 5, randomly, they're in a random order in these strings, right? And these are the 2 removal times. So we go back after 3 months and remove this half of the bag, weigh all the litter. We knew it was 8 grams to begin with, so we bring it back, dry it, say it's 7 grams, it's lost a gram, right? And then 3 months later, come back and do removal 2, and you can see how fast is mass being lost. And that's an indicator of the decomposition rate. And this is the in-stream bags. And these are attached with these really long earth staples. It can be very challenging to get them in there, but also really fun. So, what did we find? Decomposition in terrestrial habitats. This figure shows the incubation length on the x-axis, the mass remaining, so the percent of the initial weight that we put in the bag on the y, and the different color lines are the different treatments, percent of Acer platanoides, right, the Norway maple. So the blue line is all invasives, 75% invasive litter, 50, 25, and none. So we ran one-way ANOVA on these. Was it 2A? Forgive me, I forget. But nothing was significant here, except that mass is lost over time, right? That was to be expected. Although sometimes in decomposition you actually gain mass because you have bacteria and fungi latching onto stuff, so it can actually get heavier. We did see what we expected, mass loss, but there was no significant difference between our treatments, which is a little bit not great, not what we expected. But I would point out that the 100% platanoides is has the least amount of mass remaining in both situations, and 100% sugar has the most. But I say that because I have to, because she made this graph, but not statistically significant. So what do we see with the invertebrates in these bags? These are the terrestrial bags. We saw no significant difference in richness. This is a similar figure, incubation length here, species richness. So no significant differences with richness, no significant differences with abundance, no significant differences with Shannon-Wiener diversity. But what we did see when she did the Jacquard's index, coefficient, excuse me, she did get something significant here. So that was fun. And I'm going to take some time explaining this because it's different from my other figures. But these are the 2 time periods. The first time we removed the litter, terrestrial litter, second time. And what she did, she calculated this coefficient of similarity for every pairwise combination within a treatment. So how similar are all the 0% platanoides bags to each other, right? How similar are all the 25% platanoides bag communities to each other, of the invertebrate communities in those bags? And what she saw was that there was a significant difference in the similarity of the insects that are colonizing completely sugar maple bags versus completely Norway maple bags, right? And the Norway maple bags, each bag is more similar to itself than each one is more similar to its common treatment than the sugar maple bags are to themselves. Right, so if you look at this if you change the scale and you look at this on a larger scale, I would argue that you have increased diversity here, right? The different bags are less similar to each other in that same treatment when it's sugar maple. But when you have those bags, all Norway maple, each bag had a very similar community. Does that make sense? Okay, I ask my students that all the time, and they don't ever say no either. I will give you guys an exam on this to make sure. Okay, so decomposition in aquatic habitats. So these are our aquatic bags. This really struck me when Cinzia showed me this figure. I was like, wow, look at that interaction you have, because the 100% Norway maple, is crossing all these other parallel lines. And in ecology, I'm particularly obsessed with interaction figures and interactions and non-parallel lines and all that kind of stuff. But essentially, all these other bags are behaving sort of similarly and losing mass in the same way, and this one is totally like going unparalleled, doing its own thing, which I thought was interesting, and losing less mass. So this is a very different system. We're in an aquatic system now, right? And you can imagine decomposition in terrestrial and aquatic is night and day, right? There's water constantly rushing over these bags, and it appears like the Norway maples are not losing mass as quickly. Those leaves aren't losing mass as quickly as the other treatments, which have some Norway maple. So what do we see with the invertebrates here? Again, no significant difference in richness. This is a similar figure, species richness here. Shannon Wiener diversity, no difference. But this is the figure. Like, I could have just skipped everything else and just showed you guys this, but that would have been too short a talk. I got really excited about this. This shows total invertebrate abundance, okay? Incubation length here. These two lines are the ones that have all Norway maple or 75% Norway maple, right? These ones have 50% or less invasive litter in it. And there's this really clear threshold where when you have a lot of the invasive litter, you get essentially 0 invertebrates in the water. I mean, I found that really exciting. I was really excited by this. So this was our big, oh my gosh, she did get something, yay result. Or boo, depending on how you're thinking about these things. So that was a big one. So we were pretty excited about that. Again, I could have just showed you that one. I get so excited about that one. So some conclusions and future directions. These, if you recall, I reposted our initial questions. Does replacement of Acer saccharum by Acer platinoides alter decomposition rates? We don't really have much evidence for that, right? We have my one, I'll go back to this, that I was so excited about. Where did you go? But as clear an interaction as this is, this didn't come out statistically significant. So I think it's meaningful, though, I have to say, because if you look, and I intentionally put it on the same screen as this, because we're seeing there's no invertebrates. It's probably not a coincidence that they're not losing mass as quickly, right? Invertebrates are a really important part of the decomposition process. They're shredders. They're eating up the litter. They're eating up the bacteria and fungi that are on the litter. So if you don't have the invertebrates, to me, this is trending towards seeing a slower decomposition rate. That would be something I definitely am going to follow up on, although it wasn't statistically significant at the time. One reason, we only had 2 removal rates. And I would argue, given time, with no invertebrates, if we had a 3rd removal rate, we would probably see, hopefully see, or might see, increased divergence and some statistical significance there. Alternatively, more replicates, I don't know. I'm not convinced, I'm completely unconvinced that there is no difference in decomposition rate. Just from this study, I would say, I'm guessing there is. I feel like I saw a cue that there is a difference. Does replacement of Acer saccharum by Acer platinoides alter diversity and abundance of macroinvertebrates? With the Shannon Wiener index, we saw no differences in diversity. But one thing that we thought about after we put these litter bags into the Arnold Arboretum is we're working in an urban system with a potentially already depauperate invertebrate community, right? And my picture that shows potential invertebrates, actually, if you read the fine print, group 1 are organisms that are generally pollution intolerant. Group 2 are a little more tolerant to pollution. I'm going backwards, excuse me. And group 3 are organisms that are, so these ones are the tolerant ones, a little less tolerant. These are intolerant to pollution, right? So it's fairly likely that the Arnold Arboretum only started off with this group of insects, these macroinvertebrates, right? So what would be really fun would be to do this urban ecology experiment looking at gradients of urbanization or gradients of disturbance and do the same exact experiment in a really pristine, river and see if you get differences in diversity with the different litter types, right? But so that's another thing that I think would be really exciting to do. So, even with our potentially very depauperate community at the Arboretum, we do see differences in the similarity between treatments. If you recall, in the terrestrial bags, the Norway maple, so those bags were more similar to each other, right? And the sugar maple bags had a greater diversity, were more different from each other, all the sugar maple bags in their communities. And we definitely saw a really neat cue here that in aquatic environments, the type of litter really does change the abundance of invertebrates in the stream. So this is our really, I think, her big message with this experiment. But you know, it also opened up a lot of new questions and fun new experimental things we can do. So that's Cinzia, and here's thank the Simmons College, of course, the NSF, and the ecological network I work with.
You know, you just pointed out, and I'm glad you recognized it, that given the urban setting where you are, that clearly the aquatic environment could be really limited in the invertebrates it could offer. And clearly going to like just even some of the western suburbs a little bit would solve some of that.
Yeah.
I would argue too, it terrestrially probably impacted really similarly.
Oh yeah, especially where it's manicured there.
I guess, you know, given what you've done and what you've seen there, I guess the question would be, do you have any prediction with respect to either smaller microbes and the influence there, or fungal associates, and or not associates, but fungal influence, and you know, given the allelopathy that many of these plants have. Predictions there, other than the obvious, you might say?
No, you know, that's a really, really good question. I hadn't thought about that. What I've been working on now is sending it off for lignin and nitrogen analysis to see how the chemical constituents of the leaves might be influencing palatability for invertebrates, decomposition rate. But I hadn't thought about looking at the fungal or bacterial communities, which would be a really fun thing to do. And I thank you for that idea, because I could do it. That sounds like a great student project. Yeah.
If I could just piggyback on that, there have been some studies about Rhytisma acerinum, which is, you know, pretty much on every Acer platinoides tree that I've seen walking around my neighborhood. I assume everywhere else in the city. And so there has been some work on decomposition rates around that fungus as an endophyte and as a, you know, visible stromatic tissue on the leaf.
What was it? What was the name of the fungus again?
Rhytisma acerinum.
Okay.
It's the black tar spot fungus. So yeah, my earlier question was, you know, have you looked at this relationship, or if there could be any? But
no, I haven't. This is like brand new, but that's why I was so happy to bring this here, because I was trying to go through her data and think about next steps, and I'd actually put it away because I thought these folks are going to have some good ideas. So thank you. That's interesting. I'll look those up. Yeah.
I'm slightly self-conscious asking this question, but why did you choose to compare sugar maple and Norway maple? Are Norway maple actually replacing sugar maple, or are they more likely replacing things like red maple?
Right. Well, so we chose species that were dictated by Carolyn and I'm forgetting Tracy. Right. So they had set it up and made pairs for us. So
why did they choose that?
I'll ask them. I think red maple we could have done red maple. It was like compare it to one or the other, and it's just the idea is that it's whatever is locally relevant to your site. You know, I'm not able to drive students around, and so between the two pairing, it just becomes necessity. I can't legally drive Cinzia anywhere, so we had to use public transportation places, and so a lot of those decisions get made for you. But that's a good question. Just something else? Yeah.
Well, my other question is, did you look at to see if there was a difference between the lawn bags and the feral bags, or I'm assuming it's more
Oh, yeah.
the feral area than
The lawn
area.
bag. Oh, yeah. I was like, the feral bags? Good question. We analyzed it with ANOVA, so they was represented different blocks. And so with the statistical analysis we did, the variation due to blocks comes out, but so we hopefully removed that type of variation with that analysis, but we didn't consciously compare the two, which would be a really interesting thing to do. Do that when I go home. Yeah.
So I was curious about your sampling design at ANOVA too, because you've had so many results that didn't come out significant. So it sounds like you accounted for site in that ANOVA. Was that a fixed or a random effect?
That was a random effect.
Okay. And was it just one replicate of each treatment per site?
Well, two. So each treatment, so
Right.
because the two time periods.
One per
Yeah,
time period.
and each site had a paired terrestrial aquatic, but they never got compared because they're actually, we didn't compare the terrestrial to the aquatic because it's not. doesn't
Yeah,
make
I wonder
sense.
if that sampling design sort of swamping your variation at the treatment level.
In what way?
Well, you only have one replicate per site.
Right.
And so your within site replication is non-existent. And therefore
Right.
each site is essentially your error term in the model. And you're doing a random effect, so that's good because you're only using two parameters for that. But at the same time, I think you lose that random effect, or you lose an error term essentially in site. And by
Yeah.
doing that, then you're comparing site, which has a much larger influence than it should, to treatment.
Yeah.
I think just doing two replicates, or certainly three, would help.
Yeah. Well, again, that is dictated by the larger
Sure.
study, but perhaps a different analysis. You know, I go back and look at that and whether the ANOVA is really appropriate.
You might get some improvements with Bayesian analyses,
Yeah.
but I don't know if that's a route you want to take there.
Well, I wouldn't have my student take it, but I
Sure.
could go down that road. I'm not going to send Chintzia
Yeah.
down that road, though. But that's a really, really excellent point. Thank you. Yeah. Oh,
I'm sorry.
yeah.
Well, I was part of a study that was looking at wetland assessment protocols, and one of the things that we looked at was actually algae growing on red maple leaves in our various sites throughout. And you know, we mentioned the fungus, we mentioned the invertebrates. You might also look at what kind of algae are growing
Yeah.
and helping to break down. Now, you know, red maple is a wetland species, but you
Yeah.
might find some differences in your aquatics specifically with the algae.
Yeah. Yeah. Definitely. Yeah. Sorry.
In your bags, I assume that the leaves were layered. Okay. Was there any concern about how that might affect decomposition?
So it was 8 grams in each bag, and so it wasn't like 5 layers deep. She
Okay.
just, slipped them in, and so there was some overlap.
And they were whole leaves?
Yeah.
Okay.
Yeah. Yeah, so there wasn't a huge amount of layering.
Okay.
It wasn't like a sandwich. It was, but, it wasn't a single leaf either.
Right. I asked because this may sound weird, but
Not at all.
I'd take some leaves from the wild. I was looking for something else, but I'd take the leaves and I put them in my backyard. But when I first did this, I had layered them very nicely and neatly because they're leaves. And they were all rotted completely. And then another time I took some other leaves and left them more in a jumble in a big brown paper bag.
Right.
And those seem to survive better. And this is in an urban environment.
Yeah.
I don't know if it's like invasive worms in my backyard. I don't know what else is going on back there. But it makes me think about how the experiment is conducted.
Yeah.
And I just was curious if that was considered, if you had to deal with that.
These litter bags are sort of the standard way to do this, and I think that I never thought about sort of the potential confounding or issues of how they're stacked or how they're stuffed, because I could see that underground, people at different institutions could be filling them slightly differently, and that's a really good point. And to be totally honest, Cinzia did all of the filling of the litter bags, and I was there when we deployed them, but I didn't look to see, I didn't look that closely at how she put the leaves in. So that's an interesting point that I would be careful looking at in the future and think about.
It may be something that makes no difference at all.
But it might. It'd be
Something I
kind of fun
noticed
to
in
do a little litter
observation.
bag experiment where you have a different shaped litter bag, right? That allows for the leaves to be in a more three-dimensional arrangement. You can make a litter tent
Yeah.
and a litter bag and see if there's a difference.
Yeah.
Well,
That'd be
you
cool.
could be looking at invertebrate access to each
Yeah.
of the leaves. The first one they come upon is the sugar maple versus the first one they come upon is the Norway.
Norway.
It could
Oh,
be a
right.
difference.
The order of the,
And whereas they're packed right in, they're
Mhm.
not able to just pick and choose and go over.
right.
So it could be
Yeah.
tricky stuff.
It's very tricky stuff, science.
In
Yeah.
addition to the point John was making about the order of the leaves, I was thinking of it from a different perspective of if there are toxins of some sort, allelopathic agents in the Platanoides leaves, if they're on top and the rain's coming through, as is the terrestrial ones, then it might have a different effect on the bottom ones than if they're not able, if they're not overlapping or if they're not on top. So
Yeah.
there's amongst the
Yeah.
things that you have here are enormous numbers of new questions.
Yeah.
Yeah.
Well, and so what I think I failed to mention is sort of the idea behind the different ratios of litter in each treatment is sort of different stages of invasion. So you have your pristine environment, you have early invasion where it's just a quarter of the litter, you have a little bit later where it's half the litter. So that is an interesting point in that, with the different proportion of the Platanoides litter in the litter bag, potentially more toxins would be leaching out, right? If there's a greater proportion of that litter. So that, not so much to your point, but it's just that was a little bit that I think I forgot to convey to you guys earlier.
I had a question back to the point that was raised earlier, very early. And you brought it up in your talk, if it's the algae and fungi and other microbes that are on the leaves that some of the invertebrates are eating, it may be those organisms or those taxa that are affected by whatever is in different leaves, rather than the insects being directly affected.
Yeah.
Which is another set of questions,
Yeah.
and much harder to address for
Yeah.
those graduates, because you gotta get into the species of microbes.
Yeah.
What
No,
do
that
you
was
think?
something Cinzia had wanted to do, but didn't have the time. So we treat that sort of as a black box. Like, yes, that's there, and that's probably an intermediary, but we were unable to address it.
I had a very quick question.
Mhm.
Like, take up all the questions here. Has anybody looked at the rate of decomposition of Platanoides in its native habitats?
Oh, that's a great question.
Because there's so much else going on, and years ago, I mentioned to somebody that in our Norway maple forest, there's hardly anything growing underneath the Norway maple trees.
Mhm.
And whoever it was said, what grows under them at home?
Right.
And I said, oh. Are there things that are adapted?
Yeah, it's a great question.
Any harassment of the bags in, either by animals or people? Did you notice any
No, we
confounding?
were worried about that because people are walking by, the sort of the manicured sites, people are walking by. But where we put our litter bags, the Arboretum very graciously didn't mow that area while our experiment was in place, so it didn't get disturbed by management. And we picked the sites to be, maximally hidden from passersby. So I don't think I was amazed. I don't think we had a single bag disturbed. We found them all. They all came home, which was amazing.
People don't eat out of that stream very much.
I've seen dogs running around though, or raccoon.
Yeah.
And so we had some litter bags in the unmanicured part, and there's just beautiful graffiti all over the place where we put some of the litter bags. So I thought maybe people are coming here to, do all kinds of stuff.
One other question. Do you know what the vegetation in the unmanaged area was?
Oh my gosh, I do have it written, I don't, off the top of my head, I couldn't say, but.
I was more in the order of, my suspicion is that it's heavily laden with exotics itself.
Oh, yeah,
So it's not,
yeah,
you're still in the urban.
yeah, definitely.
I think if there's any more questions, we can talk about it over refreshments in the next room.
And we have an abundance of refreshments, right?